Hello friends, welcome back to my YouTube channel Nasrin Parasale. So in this video, I'm going to share with you 10 important questions which you can expect for your GATE 2023 Biotechnology paper. So first question, how many isoprene units are there in sesquiterpene? So I had given you an isoprene unit present in each terpene class. So with regarding to isoprene, they form a basic structural skeleton for all your terpenes. So this is structure of isoprene unit. They are 5 carbon containing compound. Okay, monoterpene totally 2 isoprene unit will be present. In sesquiterpene totally 3 will be present. In diterpene totally 4 will be present. And in triterpene totally 6 will be present. And in tetraterpene totally 8 will be present. Regarding the example of monoterpene include your eucalyptus, camphor, lemon and menthol, thymol. And the example of sesquiterpene include your abscisic acid. And with regarding to diterpene, it is an example of gibberellin. So abscisic acid is belonging to a class called sesquiterpene and gibberellin is coming under diterpene group. Okay, so you should know that. And in regarding to example of triterpene, it includes your saponins, prazonosteroid. The example or the group belonging to tetraterpene include your carotenoids and xanthophyll like lutein and zeaxanthin. Next question, name the secondary metabolite which is characterized by the presence of hydroxyl group with aromatic ring. Ultimately, you know the answer, it is phenol. So this is a structure of phenol where you can find aromatic benzene ring and here you can able to find a OH group which is a hydroxyl group. And flavonoid, anthocyanin and stilbenes are the example of this phenol group. In each of that you can able to find the aromatic ring and in addition to that you can able to find a hydroxyl group. So the correct answer is phenolics. So the plant secondary metabolite are classified into terpene, phenolic, N containing compound and S containing compound. So terpene the basic unit is isoprene and for phenolic the basic structure unit will be having aromatic ring along with the OH group. And the example of the group coming under the nitrogen containing compound include your alkaloids, cyanogenic glucosides, non-protein amino acids. And the example of sulfur containing secondary metabolite include your glutathione, glucosinolates, phytolactin, threonine, deficient alanines. Okay. Next thing, intermediates of which of the following metabolic pathway has not been used in synthesis of amino acid. So for which you should know the biosynthesis of amino acid pathway. So here you can able to see the ribose 5-phosphate and erythrose 4-phosphate are coming from pentose pathway, and this ribose 5-phosphate will be giving rise to histidine amino acid, and ultimately it will be forming serine. And phenylalanine, tyrosine, tryptophan will be produced from this erythrose 4-phosphate. And with regarding to glycolysis pathway, from glycolysis pathway 3-phosphoglycerate, phosphoenol pyruvate and pyruvate will be coming, and from this 3-phosphoglycerate your serine will be produced, and from your pyruvate alanine, valine, isoleucine, leucine will be produced. And from citric acid 2 intermediates has been taken. One is oxaloacetate and next one is alpha ketoglutarate. And oxaloacetate will be giving aspartate, and from that asparagine, methionine, threonine, lysine will be produced, and from alpha ketoglutarate your glutamate amino acid will be produced, and based on from the transamination reaction glutamate, proline and arginine will be produced from the glutamate. So here they are asking which of the following has not been used. Ultimately the intermediates or any of the precursor compound involving in fatty acid biosynthesis has not been involved in the synthesis of amino acid. Next question, what is the concentration of carbon dioxide required for culturing animal cell? The correct answer is 1 to 10% of carbon dioxide will be using for animal cell. Generally, most commonly 5% carbon dioxide will be generally employed. Regarding to pH level, mammalian cell generally go at a pH of 7.4. These are additional differences, friends. And fibroblast cell line, they prefer to go in a basic environment ranging from 7.4 to 7.7. And the insect cell line, like your SF9, SF21, they will be growing at a pH of 6. And regarding to temperature, your human and mammalian cell line, the cell line which are derived from human tissue or mammalian tissue, should be maintained at an average optimal growth temperature include 36 degrees Celsius to 37 degrees Celsius. And the insect cell line, they generally culture at 27 degrees Celsius because they start to grow slowly when they had culture in a lower temperature or the temperature between 27 degree to 30 degrees Celsius. So if the temperature is crossing 30 means they will be losing the viability of the insects will be used. We can't even recover them. Next, avian cell line, they require 38.5 degrees Celsius for maximum growth, and also at 37 degrees Celsius also you can able to maintain, but at that temperature they will be growing very slowly. And the cell line that are derived from your cold-blooded animals like cold water fishes, amphibians, they can tolerate a wide temperature ranging from 15 degrees Celsius to 26 degrees Celsius. Next, you should know about the optimal concentration of L-glutamide, which will be acting as an amino acid supplement for all your cell culture. It depends upon the cell type, what type of cell you are culturing and what type of medium you are going to prefer. Okay, the optimal concentration of glucose range from 2 to 6 millimolar, and it also glucose, which is a soluble sugar that will be added to cell culture media, which will be supplying the energy source. Okay, and the lowest concentration of glucose is 1 gram per liter, which is 5.5 millimolar, and the highest concentration is 10 gram per liter, which is 55 millimolar. And fetal bovine serum, they are generally added to the media, the concentration of 5 to 10%. Next question, name the organism on which first cell line was obtained. The answer is mouse. So first cell line is named as L line 29, which is also called as L cell line, and it was established by Earl in the year 1948, and it has been derived from subcutaneous mouse tissue. So here you can be able to see the name of the cell line and from which tissue they had derived, and this is a morphology. So at least know from which tissue the particular cell line has been derived. So HeLa cell line that have been derived from human cervix, and CHO is derived from Chinese hamster ovary, and MDCK is from canine kidney, and WI38 is from human lung, and same MCR5 is also from human lung tissue. And BH421 is from Syrian hamster kidney, and this virocellin is from African green monkey kidney. And your NIH3T3 is derived from mouse embryo, and the SHSY5Y is from human neuroblastoma cell line. Okay. Next is name the cyclin which take part in the mitosis phase. So cyclin ultimately, they are the regulatory subunit of your cyclin dependent kinases. So CDK are the central component involved in controlling all your cell cycle. Okay, this cyclin or CDK regulator which will be controlling the activity of CDK. So cyclin are classified into 4 types based on which phase of cell cycle they are being involved. So cyclin D is involved in regulating G1 phase, and cyclin E is involved in regulating G1 to S phase transition. So this cyclin E is responsible for triggering the cells to enter into synthesis phase. And cyclin A is involved in regulating the completion step of the synthesis phase, and next cyclin B is responsible for mitosis. And here they are asking the cyclin, which cyclin takes place in M phase, it is cyclin B, and also E cyclin will be having their respective CDK partner. If you take cyclin D, the CDK partner is CDK4 and CDK6. And with regarding to cyclin E, the CDK partner is CDK2, and for cyclin A, the CDK partner is CDK2 and CDK1, and for cyclin B, the CDK partner is CDK1. Next, with regarding to coronavirus, so you will be expecting coronavirus question, either they will be asking genome or they will be asking vaccination for this coronavirus. Okay, so here coronavirus have a positive sense single strand RNA and belong to a family called Coronaviridae and order called Nidovirus. And the size is ranging from 26 kilobase per to 32 kilobase per, and currently 4 variants have been identified, and alpha and beta have been more prevalent in mammals, gamma in apes, and delta in both mammals as well as apes. So here I had given you additional reference. Example of single-stranded DNA containing virus include your parvovirus, M13 fudge and M12 fudge, and those viruses with double-stranded DNA include your adenovirus, herpes virus, pox virus, plant virus, cauliflower mosaic virus, and your T fudge virus and lambda fudge virus. Next, those viruses with single-stranded RNA include your animal virus, paramyxovirus, orthomyxovirus, tahavidovirus, TMV virus, turnip yellow mosaic virus. The TMV, tobacco mosaic virus, is having a single-stranded RNA, whereas the cauliflower mosaic virus is having double-stranded DNA. And those viruses with double-stranded RNA include your reovirus, retrovirus, plant virus, rice stalk virus. Okay. Next question, which of the following organelle is surrounded by a single phospholipid layer? So here I have given you a detailed understanding. So single membrane organelle include your lysosome, vacuoles, Golgi body, endoplasmic reticulum, microbodies like peroxisome, glyoxysome, glycosome, hydrogenosomes, magnetosomes are having a single membrane organelle. They will be having a single phospholipid layer. So here Golgi apparatus and lysosome, both options are correct. Then example of those organelles who are having a double membrane organelle include your mitochondria, chloroplast, nucleus, and plastid. Next, some organelles are considered as a non-membrane bound, which means they don't have any membrane around them. Example include ribosome, centromere, inclusion body, nucleolus. So the nucleus is coming under double membrane organelle, whereas nucleolus is coming under non-membrane organelle. Next is cilia and flagella, microtubule, microvilli, cytoskeleton, and centrioles. Okay, so these are non-membrane bounded organelles. Next, they will be asking one question from autoimmune diseases. So it might be a match the following question. In this question, they are asking match the following autoimmune diseases with the location of self-antigen. So I think you can find this particular PDF or tablet or column in a QB book. So all autoimmune diseases are classified into organ specific and systemic. The example of organ specific include your Addison disease, autoimmune hemolytic anemia, Goodpasture syndrome, Graves disease, Hashimoto thyroiditis, idiopathic thrombocytopenia purpura, insulin dependent diabetes mellitus, myasthenia gravis, myocardial infarction, perinicus anemia, pro-streptococcal glomerulonephritis, and spontaneous infertility. And those autoimmune diseases which are considered systemic include your ankylosing spondylitis, multiple sclerosis, rheumatoid arthritis, scleroderma, Sjögren's syndrome, systemic lupus erythematosus. Okay, so again they will be asking this particular question also. Now we need to know a self antigen for the respective disease. If you take Addison disease, the self antigen is your vaginal cells, most commonly cells of vaginal cortex. Okay, in autoimmune hemolytic anemia, the self antigen will be located on the RBC membrane protein, whereas in your idiopathic thrombocytopenia purpura, the self antigen will be present on platelet membrane protein. Next, in Goodpasture syndrome, the self antigen is your collagen. So in a Goodpasture syndrome, the self antigen is your collagen, which means the collagen will be most commonly present in the basement membrane of your renal and lung tissue. Next, in Graves' disease, the self antigen is located on thyroid stimulating hormone receptor, whereas in Hashimoto thyroiditis, the self antigen will be your thyroid protein and your thyroid cells. Next, in insulin dependent diabetes mellitus, self antigen will be located on the pancreatic beta cell. Next, in myasthenia gravis, the self antigen will be located on acetylcholine receptor, whereas in multiple sclerosis, the self antigen will be in your brain or white matter. Okay. Next, in myocardial infarction, self antigen will be in your heart, and in Peyronie's anemia, the self antigen will be either in your gastric parietal cells or as intrinsic factor. Okay. Next, in post-streptococcal glomerulonephritis, the self antigen will be present on kidney, and in spontaneous infertility, self antigen will be located on sperm. Next, with regarding to this ankylosing spondylitis, the self antigen will be located in your vertebrae. Next, in rheumatoid arthritis, self antigen will be located in the connective tissue, or else the self antigen will be your IgG antibody. Okay. Next, in scleroderma, the self antigen will be located either in the nucleus, heart, lung, gastrointestinal tract, and kidney. And the Sjögren's syndrome, the self antigen will be located either in your salivary gland, or thyroid gland, or liver, or kidney. Next, in systemic lupus erythematosus, self antigen will be your DNA or protein or RBC or platelet. So your immune system will be fighting against your own DNA, nuclear protein, RBC, and platelet membrane. Okay. So based on that, if you answer this question, Graves' disease, it is thyroid cell, rheumatoid arthritis is IgG, and type 1 diabetes mellitus, it is pancreatic beta cells, and for Addison disease, self antigen will be located on the adenosine. So the last question is that a positive tuberculin test is an example of, this test is an example of delay type hypersensitivity, which is also called as type 4 hypersensitivity or cell-mediated hypersensitivity. Again, even though you can't able to refer of everything, at least study the tablet or column. The type 1 is mediated by IgE, and type 2 is mediated by IgG, and type 3 is mediated by immune complex, and type 4 is called the cell mediated. And the typical manifestation of this type 1 include your anaphylactic reaction like your hay fever, asthma, hives, food allergies, and eczema. And for the typical manifestation mediated by this type 2 include your blood transfusion reactions and your erythroblastosis vitellosus and autoimmune hemolytic anemia. The typical manifestation of this type 3 include your arthritis reactions and serum sickness, necrosis, vasculitis, glomerulonephritis, rheumatoid arthritis, and systemic lupus erythematosus. And the typical manifestation of type 4 is contact dermatitis, tuberculin lesions, and graft rejection are coming under type 4. So this tuberculin test is an example of dilated hypersensitivity where they will be injecting the tuberculin fluid, which is nothing but it's a filtrate from the tuberculosis causing bacteria, Mycobacterium tuberculum. They will be injecting in the lower part of the arm, and they will be giving a time point of 24 to 72 hours. Okay, because it takes time to develop a particular hypersensitivity reaction, that's why it's called as hypersensitivity reaction. And based on the size of swelling, they will be diagnosing the TB. If the swelling size is 5 millimeter, means the person is in close contact with the TB patient, or for a healthy person, the size of swelling on induration will be little bit high. That is, the size of swelling is 15 millimeter, means the person is a healthy person. So this is a tuberculin test is most common test. The foundation was laid by Robert Koch, who had initially worked in this particular tuberculosis causing bacteria. Okay, so have a brief understanding about this tuberculin test also, in addition to that hypersensitivity test also. Because this for tuberculosis, they will be going for two tests. One is TB skin test, another one is TB blood test. The tuberculin test is coming under TB skin test. So soon I will be uploading a part 2 video also. Thank you friends, thanks for watching this video.